All right, there you go. Names and class periods. Thank you. I'll leave that open in case other people come by. Thank you. You're welcome. Thank you. All right. Afternoon, y'all. Nice to see you. How you guys doing? Cool. All right. Today, this is our 4th out of 6. We're 2/3 done after this review session. And our topic tonight is the DNA stuff. I'll also throw in a little bit of electrophoresis. And we've also got a pretty good practice problem to work on. And yeah, so anyway, transcription, translation, and gene regulation is the stuff that seems like we've kind of come back to more and more and more. Like I feel like I've reviewed it 50,000 times already. But it is something that's pretty big on the AP Bio test. Question 6 on the free response over the last 4 years has always been molecular genetics. So molecular genetics is like transcription, translation, all that, as opposed to like classical genetics. That's like Punnett squares and Mendel and big A, big B, little b, that kind of thing. Okay, so first off, electrophoresis. What was it? So talk with somebody around you. I've got 2 questions you're going to try to figure out. We had that kind of clear stuff. Does anybody remember what that happened to be called? Started with an A. Yeah, agarose. Yeah, so I have agarose, and it is similar to agar. So those of you guys that have had microbiology or like from that bacteria lab, that was agar. Agarose was slightly different. So my first question is, why did we use agarose? What was so special about that? And then my second question was, what does electricity have to do with any of it? Like, why was I hooking up this gel to electricity? So, why agarose and why electricity? You could say like genetics, yeah, or molecular genetics, yeah. All right. So somebody raise their hand. Pick whichever one you want to answer. Somebody tell me what you think about one of them. Yeah, Kristen. Mm-hmm. Good. So agarose is cobweb-like. So if I look at my gel from the side, then remember we kind of had like those little, they call them gel combs that were in there. So I put the gel comb in the gel box, then I pour the agarose in there, and then when the agarose solidifies, it was kind of jello-y like. Then if I take the gel comb out, then there's a whole bunch of like little divots, and we call those wells. And so you're exactly right. My agarose is cobweb-like. And why do I want that? Like, why is that a good thing? Yeah, Eleanor. Yeah, DNA can run through to the other side. What if it wasn't cobwebby? Couldn't it get through easier? Yeah, so why do I want it to be cobwebby? Uh-huh. Yeah, different lengths of what? You're exactly right, but yeah, different lengths of DNA go different distances. So if I have all these cobwebs in here, if I make it super concentrated, it'd be a little bit more cloudy and it'd be extra cobwebby. And then if I make it like more dilute, then it's less cobwebby. So I can decide how strong or how weak to make my agarose based off of what I think is going to be traveling through it. So if I have giant hunks of DNA traveling through it, I might make it a little bit less cobwebby. If I have really, really small chunks of DNA traveling through it, I think, then I might make it more cobwebby. And so what will happen is if I've got a piece of DNA that was cut, what chemicals is it that cuts DNA again? Huh? Yeah, restriction enzymes. You might hear it restriction endonuclease. All right, so if I cut my DNA, if I have a piece of DNA that's that big, one that's that big, and one that's that big, which one is going to travel, and I fill it up in this well right here, which one is going to travel to the end the fastest, the small, the medium, or the big? The small one, that makes sense. Things that are physically small could probably like weave through these cracks easier than something that's really big. And so that's what's going to happen. By the time I get to the end of the gel, the small one might make it like this far, and maybe the medium one is like this far, and maybe the large one is like that far. And that's why there's like a distinct banding pattern. So from the side, it looks something like that. I get it to travel through there. Why electricity? Why shock this? Yeah, Caleb. Mm-hmm. Yep, you're exactly right. In DNA, the backbone of DNA has phosphates. Phosphates are negative. Again, seems like that comes up like once a week in class, the fact that phosphate's negative. And so if I can put this in a gel, and if I face it the right way, if there's a positive wire over here, then my DNA is going to chase that positive wire because DNA is always negative. Your DNA, a dog's DNA, an apple's DNA will always travel towards the positive. What if I messed up and I put my gel in backwards? It would still run this way, but now, wouldn't I be loading my gel right here? Wouldn't it like run like, I don't know, a centimeter and then just like leave? Yeah. So the reason I have electricity is because the positive pole attracts the negative phosphates. And then the reason I have that webbing is so now if everybody is taking off and they're chasing towards this positive pole, small pieces weave through faster, medium pieces slower, and then large pieces the slowest. And so eventually you get like that kind of unique banding pattern. And they sometimes might call this like a DNA fingerprint or something like that. Let's say that I was doing genetics problems. This is kind of tricky. I've got a person and I'm checking just these pieces of DNA. I'm going to load this in well number 1, well number 2, and well number 3. This might be something that like seems like a valid question in my head, but I might not be giving you enough information. Which of those do you think will have 1 band, and which of those do you think will have 2 bands? You can talk with somebody around you. So like lane 1, 2, 3. Some of them will have 1 band. Some of them will have 2 bands. Some of them have thick ones. Some will have thin bands.
You know what? That's a great thought. We're going to run with that one.
Okay, so I'm going to re-ask it. It's 2 questions. Which ones will have 0 bands or 1 band or 2 bands, like number of bands? And then will those bands be thick or thin? So for like each band you talk about, is it thicker or thin? So the option is either it's a thick band or it's a thin band. And it's either got 1 or 2. So what do you think, Sarah? Yes, the heterozygous will have 2 bands. Why? Yeah, 2 different types of DNA. All right, now, next question, and anybody can take this. It doesn't have to still be Sarah. It can still be Sarah. But thick versus thin. So how many bands will this one have, first of all, 1 or 2? 1. How many will this one have? 2, because these are different pieces of DNA. How about this one? 1. This band over here, thick or thin? Thick. Why is it thick? Are dominant bands always thick? That was funny. Like 6 of you guys said that. You're like, because it's dominant.
Like the dominant, like things that would be like thicker, a little bit.
But recessive doesn't necessarily have to mean that it's smaller. It just means that it's recessive, right? Like the gene for blue eyes might be bigger than the gene for brown eyes. Blue eyes might exist because we have a brown eye gene and we had an insertion of DNA, so it actually is broken because it's bigger. Huh? Or actually, let me do something really quick. So maybe I have this. All right, so comparing these three bands, do you guys think that that one is thick or thin? Thick. What about these two? Thin. Why is it logical that those are thin and that one's thick? I've got twice as much DNA in one spot. If I stain DNA purple and I put in 10 microliters in there and 10 microliters there, and 5 microliters of DNA that's painted purple stops there, and 5 microliters stops right there, then it's a certain darkness. If 5 microliters stop there and the other 5 start right there, then that's 10 microliters, so that's like a darker purple band. Does that make sense? If I put down 5 microliters of blue ink and 5 microliters of blue ink, that's lighter than if I put down a line of 10 microliters of blue ink. So that line will be thicker and darker. That one will be thinner. How many bands will this one have? 1. Will it be dark or light? Or like thick or thin? Thick. And where will it be located? On the top or the bottom? Bottom. So what letter is that one representing? Big B, yeah. So how far does big B travel? That far. What if I have 1 amount of big B? It's that dark. What if I have twice the amount? It's going to be darker. How far did little b travel? That far. So that's little b, so it would have had to travel that far. So I can now go through and say, that's little b, that's a little b, that's a big B, and that's a big B and a big B. All right. The rest of the stuff on electrophoresis probably doesn't matter, getting into stains and all that. That's not going to show up on the AP Bio test. So they generally want to know like how stuff works or why it works. They don't get into like how many milliliters did you pour in or something dumb like that. Okay, so any questions on gel electrophoresis? Cool, and what was it called again? I know I asked it a few minutes ago, but what was the name of that chemical that cuts DNA at specific spots? Yeah, a restriction enzyme, or they might call it restriction endonuclease. All right, cool. So now let's try a practice problem. I know. So question number 6 on the free response for the last 4 years has been molecular genetics. Question number 6 on the free response almost always feels like it's too long to be the last one. So it always looks, you're just like, you're sitting there, you're getting to the end, you're like, I'm running out of time, and you're like, it's the last one, certainly they'll be nice, and I'll be like, you mother, and it's just like paragraph, paragraph, data table, maybe another graph. However, question 6 is usually not bad. It's just, do you have time to trudge through it? Okay, so this one I don't have copies of. So for you guys, it might be easier to take a picture with your phone and then be able to like look at it. If you wanted to look at it later, or if you're watching the video and you wanted to like find it, it's 2022 number 6. But we'll read this part and then we'll like look at this data table. So take a second and read it. Do you guys want me to read it to you? Biology story time? Yeah. Biology story time. Just like when you were a little kid and you'd read like, did you guys ever read Goodnight Moon? This is exactly like Goodnight Moon. Researchers are studying the use of RNA vaccines to protect individuals against certain diseases. Let me switch cameras here. There you go. To develop the vaccines, particular cells are first removed from an individual. Great. Then mRNAs coding for specific proteins from a pathogen are introduced into the cells. Great. The altered cells are injected back into the individual, where the cell makes the proteins encoded by the introduced mRNAs. Cool. The individual then produces an immune response to the proteins that will help protect the individual from developing a disease if exposed to the pathogen in the future. Turns out I don't need any of this when they start asking me stuff. So everybody's a little bit different. Sometimes students ask me, hey, Mr. Cotton, do you recommend that I read the questions first before I read all the background stuff? Maybe you skim through it. If you want, it all depends. So whatever you think saves you time. For me, I kind of read everything, try to process it as I go, and then hop into the questions, and I try to not have to read it again. But everybody's a little bit different. All right. When introduced in the cells, the mRNA used for the vaccines must be stable, so they are not degraded before the encoded proteins are produced. Okay, so I need it to be stable. Researchers developed several modified caps, I'm thinking 5' caps, if I'm you guys, that they hypothesized might make the introduced mRNA more stable than mRNAs with the normal GTP cap. GTP cap is the 5' cap. To test the effects of the modified caps, the researchers produced mRNAs that differed only in their cap structure. These are all the different versions. They introduced the same amount of each mRNA to different groups of cells and measured the amount of time required for the half-life of the mRNAs to degrade. That's mRNA half-life. And the total amount of protein translated from the mRNA. Okay, so I have half-life, total amount of protein translated from the mRNA. All right, cool. All right, so let's try part A first. Based on the data, identify which cap structure is most likely to protect the end of the mRNAs from degradation. On your own, with somebody else, whatever helps. And real quick, is the sign-in sheet still making its way around? Where is the sign-in sheet? Oh, okay. Did we miss anybody? Okay, so we'll pass it kind of back that way, and then whenever it gets to maybe Samuel back there in the back, it looks like some of these people over here need it. All right, so that everybody can kind of vote, we've got 5 options. So you got option 1, so we'll just be 1. You got option 2, 3, 4, and 5. So 1, 2, 3, 4, or 5. What do you guys think it is? Option 1, 2, 3, 4, or 5. It's option 4. Why? It lasts the longest. So half-life, anytime they use the word half-life in science, that's how long it takes for half of something to break down. Normally you hear about it in chemistry when we're talking about carbon-14 degrading over time and its half-life is 5,000 years or something like that. So here it's how long does it take for half of the mRNA to break down? So modified Cap2 lasted the longest. How do I know what's going on here? Measure the amount of time required for half of the mRNAs to degrade. That's mRNA half-life. So half-life is telling me how fast does it degrade. Now, I did kind of have to know what was going on based on identify which structure is most likely to protect the ends from degradation. I did have to know that half-life was degradation. So I probably did, for a lot of students, I probably did have to read that to answer this. All right, B. Based on the data for the mRNAs with modified caps, describe the relationship between the mRNA half-life and the total amount of protein produced. Relationship between mRNA half-life and the total amount of protein produced. All right. Anybody want to? Thank you. Did we miss anybody on the sign-in sheet? Cool. And I usually get those in within like 2 days, I would say. Tomorrow I get a conference period, so I'll probably get them in tomorrow. So if you were to look at like ones from last week or the week before and like your name's on there and I didn't give you credit, let me know. So usually give me about 2 days to get those in. And if it's like, hey, that one's a week ago and you didn't have it on there, let me know. I'll just go back. I'm sure I overlooked you on the sign-in sheet and I'll fix it. So what do you guys think? What about the relationship between the mRNA half-life and total amount of protein produced? Alex?
The longer the 5' cap lasts, the more protein you make.
Yeah. You might say it several different ways. The longer the half-life, the more protein is translated. That's full credit. You can say they have a positive relationship. As one goes up, the other goes up. They have a direct relationship, and it's going up. Anything like that, and you've got it. Technically, I wouldn't have known it, but technically, I can probably answer that without knowing anything other than like reading the data table. All right, part C. After examining the data on mRNA half-lives and the amount of protein produced, the researchers hypothesized that each mRNA molecule with modified cap1 was translated more frequently than was each mRNA molecule with the normal GTP cap. Evaluate their hypothesis by comparing the data in Table 1.
That's not for me. I know.
Wait, I'm looking deep. Oh, yeah.
All right. So, anytime they say evaluate, if they like put a hypothesis out there, then you always want to say like you either agree or you don't, or like this supports their hypothesis or it doesn't. So begin by saying sort of like, don't just go into like, here's what's going on. Be like, yeah, the evidence backs them up, or like, no, evidence is against them, something like that. So do you support them or not? Yeah. And then I should say why, and it's like, well, protein translated is 4.7 plus or minus 0.042 compared to 1 plus or minus 0.007. So I would say something like, yep, evidence supports the researcher's hypothesis, and 4.777 plus or minus this is significantly higher than 1. So what if the plus minus overlapped with the plus minus here? Do you know what I'm asking? Like pretend that this was 4.7 plus or minus 0.6, and this one was like 5.4 plus or minus 0.3. So take a moment and then tell me, think about how you would answer it. Like, what words would you say? You might be like, I know the answer, but like, think about what would you actually write down. They get kind of picky on this.
Like they can, but it also
All right. So they're not super picky on how you say that you like support them or not. We try to avoid saying prove, like that we prove you're right or we prove that you're wrong or something like that. But however you say, like we support or we don't support their hypothesis, they're not super picky about that. But I would want to say something like 5.4 plus or minus 0.3. I could say something like the standard error of the mean, the sex, overlaps with this number and this number. I could draw something to where I'm like, hey, the range of the first one is 5.1 to 5.7. So I could be like, the range of that one overlaps with the range of this one. So this one, 4.1, all the way to 5.3. I could say the range of the first number and the range of the second number, these guys overlap, and therefore there is no statistically significant difference. So you want to say like no significant difference. Or if they didn't overlap, then, and you were having to talk about it, you could say there is a significant difference. So if the error bars overlap, then that data is considered not different enough statistically. And then again, they don't always tell us how picky they are on the way that you say it, but it seems like they're kind of picky. So again, I would say something like, if the error bars overlapped, I'd be like, the range for this number is this. The range for this number is this. Because 5.3 overlaps with 5.1, there is no statistical significant difference. Something like that. And then hopefully they aren't super picky. All right, D. Introduction of mRNAs. Actually, I don't think we need the data table at all for this part. So introduction of mRNAs into cells allows cells to produce foreign proteins that they might not normally produce. Explain why the production of foreign protein may be more likely from the introduction of mRNA than DNA into cells. Mm-hmm. Oh, I just picked that number. I was like, pretend that this was a 5.4. I was just giving you brand new numbers to where they overlapped. The other ones, they very clearly didn't overlap because one was like a 4.7 and one was like a 1. So I just invented that number. Does it make sense now? Okay, cool. If you're just like, where did this 5.5? I just pulled it out of my butt. All right. What do you think? What's the advantage of using mRNA versus like DNA? Yeah, Chris. It's more precise. How so? What's less precise about DNA? Oh, is it what you're saying isn't incorrect? But like, for example, pretend that we were new to all this, and I was like, you should use RNA, and somebody was like, why? And I'd be like, it's more precise, and you'd be like, what do you mean? Like, what's more precise about RNA versus DNA? Like, CCC is GGG, and that seems to be just as precise. What if it was from a eukaryotic cell? What if we were going to make human insulin, and we wanted to splice it into a bacteria to get bacteria to start making human insulin, and we'd call it humulin, and then that's where all human insulin would come from. So that's what we do. So why do we use RNA instead of DNA? Yeah, all human insulin is manufactured by bacteria now. They used to grind up pancreas. There's not enough pancreas in the world for all of us diabetics. I'm not diabetic, but like us as humans.
Is it because like it undergoes like all the processes, like detail?
Yeah, all the processes. So, for example, let's say that I have, I don't know, some piece of DNA from a different creature, and you give me just the DNA. Is it possible that I might splice this one way to make one thing, or a different way to make something different, or a different way, alternate splicing? So I need to have the mRNA already because you had to splice it ahead of time. If I gave you a piece of DNA and I was like, here, read this, but it hasn't been spliced yet, you might not know what you're supposed to splice. And it might be, hey, you could splice this 30 different ways to make 30 different products, but I want you to make this product. So if it's already mRNA, it's already spliced, it's got a cap, it's got a tail, and so that is better than just putting in the DNA. The DNA might work, but the mRNA is more likely to work because it's already done all the extra steps of processing. That kind of makes sense? Now, we have introns and exons. Which ones were the parts that we kept, the exons or the introns? Exons. So we cut out introns. Why is it logical, even before we checked, why was it logical that we expected, once we like discovered, pretend that we discovered exons and introns just 25 years ago. Why is it logical that scientists expected that bacteria would not have introns? You can talk with somebody around you. So pretend that exons and introns are a pretty new discovery, like 25 years. Why would scientists expect, and it holds true, it is true, bacteria in general do not have introns. When I was in high school, we had never come across any bacteria that had introns. We've since come across a couple. So why did we expect bacteria to not have introns?
They're like single stranded. One bit of word.
Make that bigger. All right. Anybody want to take a crack at this one? Why does that make sense? Why does it make sense that, hey, bacteria probably can't have introns? Huh?
Because prokaryotes, like, the DNA is like in a circle, like one gene codes for all.
That's true. Their DNA is circular, but we could still do splicing and stuff even if ours was circular. So you're on the right path. You're trying to find what's different between prokaryotic and eukaryotic DNA, and you're right. One of the things is eukaryotic DNA. Eukaryotic DNA is double helixed and then in just big long fibers until we pack it down, like in prophase, for chromosomes. And then bacterial DNA is still double helixed, but it's just joined at both ends. See, Alina? And so it's circular. It's still a double helix, but it's just that end and that end joins. So it's not that. It's something else. See it? Huh? They don't have a nucleus. What happens inside of our nucleus? That's true. We protect DNA. Once we transcribe it into RNA, then what happens after we make it RNA? We splice, 5' cap, poly A tail. So we splice. Okay, so is it possible that we make an entire piece of RNA before the ribosome ever reads it? Yes, in fact, that should always happen. So I completely make my piece of RNA, I do all the splicing, everything's ready to go, and then my final copy leaks out. Prokaryotes don't have a nucleus. So for us, our DNA, we transcribe it into RNA. So that'll be represented by this purple strand right here. And then we would process it, cap, tail, splice. Thank you, spliceosomes, for splicing out the introns, like you guys told me, and keeping the expressed exons. And now that RNA leaves, and a ribosome can land on there and read it. Okay, cool. So in a prokaryotic cell, here's my DNA. I start transcribing it. I'm not done yet. So I'm still transcribing my RNA. Is there any reason why the ribosome can't go ahead and land on this and start reading it? No, it can't. It's floating around. So in us, transcription happens first. It's totally finished. Then we edit, process whatever word you want to use. And then third is translation. In bacteria, transcription and translation happen simultaneously. As soon as I've transcribed 2% of my piece of RNA, a ribosome lands on there and starts reading it. So why can't this guy have introns? It's reading it before we ever would have even gotten a chance to splice it. So the nucleus, we learned earlier in the year, is there to protect my DNA, but it's also an editing room. So now I can do all this intron, exon, alternate splicing bullcrap. They don't. So they just read it as is. So they don't have introns. So what that means is, let's say I have a gene for human insulin. It's got introns, exons, everything in it, and I injected that into a bacteria. It won't splice it because it doesn't splice. So we don't give them human insulin DNA to make insulin. We give them the piece of human mRNA that codes for insulin. And then that ribosome will read it just fine. So we currently make, like I was saying before, it's called humulin. Think anybody, and you don't have to say that you are if you care, but nobody in here is type 1 diabetic. All my type 1 diabetics are not here. But anyway, if you have a friend, do any of y'all have a friend that's a type 1 diabetic? They have to like take insulin. The insulin that they take is called humulin, and it's manufactured by bacteria because we took the human insulin gene, got it from a cell as mRNA once it was already spliced, and then we put that into bacteria, and so now they read it and they make human insulin. That's because the genetic code is universal. So CCC codes for proline no matter what kind of plant or animal you are. AUG codes for methionine no matter who you are. It's universal. All right. Questions on anything that like popped in your head so far? Yeah.
When they manufacture the human into the prokaryote, they continue to produce that. Like, is it a negative feedback?
I don't know what they have to do to like encourage it to do that, or like how can they put it in there. I'm gonna guess, just with the knowledge that you and I would have, I'd be like, they put it in there, and I bet they put on like the longest poly A tail that is allowable, and so now we'll read it as many times as possible so that we can make as much insulin as possible. And then I don't know if they do anything else to like make it more likely to read that piece of RNA, but as long as the RNA is around, ribosomes should just randomly read it. I shouldn't have to tell them. So if I put in a piece of RNA right there, ribosomes will just automatically start reading it. If a virus infects me and it injects its piece of RNA in there, like the flu, my ribosomes just land on it and immediately just start voraciously reading it and start reproducing flu inside of my cells. They read whatever code I give them. My computer will just read whatever program I click, and it just reads that program as fast as it can. So as long as I tell it, read the program, it does. So I shouldn't have to, like, tell it, if that makes sense. Yeah, I'd probably put on a super long poly A tail, and then now I can make as much insulin as possible. So they basically just have these giant vats, and all this juice falls off, and that juice is insulin. Super weird, isn't it? So, all right, there's probably more finesse than that, but at the raw level, like, that's what's happening. All right, cool. So let's say that we've got some pictures. I'm trying to think of, like, different ways to where I'm not always saying something the exact same way. So sometimes they'll just say, explain this picture. So take a moment, think about it, explain that picture. All right. So part one is just literally explaining the stuff that I can tell by looking at the picture. And then part of it is going to be like, hey, so now that you know what's kind of going on, like, why does this matter? So first of all, we'll start with just what's happening in the picture. So I'm adding acetyl groups. And where's the acetyl group getting added to, the histone or the DNA? The histones. And then why is it that adding acetyl groups to histones makes them, like, release the DNA? Huh? Yeah, it changes their charge. Or it might even be a little bit better if I asked, like, why do they hold on to it originally? Their charges. So what about DNA is attracted to histones? Like, what about the charges? Like, what are the charges? Negative and positive? Who has the negative? DNA. What on DNA is negative? Phosphates. And then histones have positive charges. So if I add acetyl groups onto those positive charges, it'll, like, kind of like push the DNA out of the way. And now I'm kind of covering up the positives. And so now that negative DNA isn't really attracted. And so they kind of let go of each other. And why would I do this? Like, why is this significant? Like, oh, I'm getting my DNA and histones to let go of each other. What am I getting ready to do? Oh, I thought you had your hand. What am I getting ready to do? Yeah. Yeah, transcription. Yeah, about 3 of you guys said at the same time, transcription. So I just turn this gene on. Why is it logical that I can't do transcription very easily over here? Yeah, I can't read DNA when it's packed. Whenever I do transcription, remember, I've got to be able to, like, kind of like uncoil it and unzip it like this. And so if it's wrapped up super tight and it's got all these, like, protein arms hanging over the edge, I can't really do that. Whereas if I let go of it, then now I have a shot at, like, unzipping this DNA. So what we'll say is if I add acetyl groups, I'm more likely to be turning DNA on. That's just one more thing that says I'm more likely to turn the DNA on. Okay, what was another thing, see, that I could do to turn genes on and off that's on my DNA? Like, in the same unit, we talked about acetylating histones, and then we talked about doing something to DNA. Maggie? Yeah, I could do DNA methylation. Where specifically do I put the methyl groups on DNA if I want to turn a gene on or off? Yeah. The promoter. Why does it make sense that it would be the promoter? Why is that a good logical spot? Yeah, that's where RNA polymerase lands. That's where transcription starts. So if I add acetyl groups to histones, am I more turning genes on or turning genes off? Acetyl groups to histones, on. Because when I add acetyl groups, they let go of each other. So this DNA is now more in the on position. When I add methyl groups to promoters of DNA, is that DNA more on or off? Off. So if I've got DNA right here, and here's a promoter, there's my termination sequence, and maybe I've got a regulatory sequence nearby, then RNA polymerase is going to land on my promoter, like you guys were saying. But if I have a methyl group in the way, then now RNA polymerase can't land there as easily. What is a methyl group? Water's H2O. What's a methyl group? Yeah, CH3. Yeah, CH3. For reasons that don't really come up at all in this class, but just so I'm accurate in case you take genetics one day, we methylate both sides. So like if I methylate this side, I methylate this side. So I always methylate in pairs. Later on, through things that we don't really get into in here, that becomes very important. It's how you inherit your epigenome from your parents. If we only methylated one side, you would not be able to inherit your parents' methyl patterns. So, okay, so I methylate my promoter, and that turns genes off. What else could I ask that I don't ask all the dang time? I'm running out of things to ask you guys. You guys just know everything. Oh, I don't need every single detail. They're not going to ask you specifically about, like, you have to know this disease. They might bring up a disease, but they would give you enough information in the question. They might be like, this is muscular dystrophy, and muscular dystrophy, blah, 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 blah. What's the following? This or that. What was the deal with this again? Yes, there was Angelman and Prader-Willi syndrome. And what was fascinating about it, like what was so unusual, and instead of it just like, oh, it's just like another freaking disease like every other disease that's horrible. Yeah.
It was the same ones, but like different sizes.
Yeah, the exact same mutation. So I'll look at a gene. I don't remember what gene number it was. I always memorize that right before class so I look smart. But I think it was gene like 11 or 17 or, I don't know, somewhere between 1 and 23. And so on this gene, there was this certain spot that was deleted. And what was fascinating about Prader-Willi syndrome, Prader-Willi is this boy, versus Angelman, was the deletion was from the exact same chromosome. It just depended on whether it came from mom or dad. And in the end, the difference is that your body will put methyl markers on different pieces of DNA depending on whether they come from your mom or come from your dad. Most genes are not like that. Your eye color doesn't depend on did you get the capital B from mom and the little b from dad, or the capital B from dad and the little b from mom. So most genes, it doesn't matter which parent you got them from. But there are a few that it does, and they call it imprinting. The word imprinting shouldn't show up on the AP Bio test unless they tell you it there and give you the information and tell you what it is. But some genes are marked, they're methylated differently based on whether those genes come from mom or dad. Again, most genes, it doesn't matter. So for my eye color, again, it doesn't matter if I got the big B from mom or dad, I'm going to have brown eyes. But some genes, it does. What about telomeres? Why do I need those? Yeah.
Near the end on DNA, every time the cell replicates.
Yeah, I've got DNA, and every time I replicate my DNA, I lose some genetic material on the ends. Well, that's bad if those are like genes that are useful, so I just put some gibberish crap on the ends. Doesn't matter what it is. It happens in humans to be TTA-GGG usually, or TTG-GGG. But as long as it was just filler of any kind, then it's doing its job. So every time my cells divide, I lose a little bit of DNA on the ends. And so telomeres are there to make sure that I don't lose any useful DNA. Once telomeres get short enough, then my cells the current theory is once they get short enough, my cells quit dividing, and that's it. And that's why you can't live forever. We do know that when you're like 1 month old and you're still growing, like you've been on Earth, like you've been born for a month, not like mom's 1 month pregnant, but like you were born last month, you're here on Earth, and we do know that when you're like 1 month old, you have telomerase. And so telomerase keeps rebuilding your telomeres for the first year, year and a half, 2 years of life. So it's very possible that in the near future, we'll find a way to turn telomerase back on. And as long as we can turn telomerase on and off in the right cells, then we could extend lifespan for reasons that we don't necessarily understand. When telomeres are short, cells start acting like they're old. When we make those exact same ones longer, those cells start acting young again. Now, what I don't want is if I've got a cancer cell right here, I've got like, I don't know, some sort of kidney cancer. If it starts to grow and it runs out of telomeres, that's awesome because now that cancer can't grow anymore, right? So cancers will sometimes start to grow. They get to maybe 1 millimeter in size and then they're stuck because they run out of telomeres. So you just avoided having a giant tumor take over your body. But if that cancer then mutates and now telomerase turns on, then it can start growing out of control again. So we think telomeres were there originally so that cancers can't grow forever. But if telomerase is on, then cancer cells can grow forever. Yeah?
Are telomeres shorter on animals because they don't live as long as
no, so we don't think that we're currently dying from lack of telomeres. So we die from cancer, heart attacks, accidents, stuff like that. So we don't think that like currently my dog is dying. Like if I have a boxer, my boxer's going to die from cancer because that's what boxers die from. If I have a German Shepherd, it's going to blow out a hip. They have hip dysplasia. So we die from something else before we run out of telomeres. Yeah, we're done. Yeah. Enzymes can build stuff up or break stuff down. So like, for example, you, when you were 1 year old, you weighed however many pounds, and now you're an adult. What built you up? Enzymes. So enzymes can build up or break down. Oh, thanks.